Welcome back to part 2 of our TPD webinar series on targeted protein degradation, titled Molecular Glues from Serendipity to Rational Discovery. My name is Dimitrios and I'm the CEO of Taros Chemicals. Today's session focuses on molecular glues, a pioneering class of compounds that are pivotal in the field of targeted protein degradation. These molecules originally were discovered through fortunate accidents and are now being strategically explored to harness their full therapeutic potential. At Taros, we're excited to be at the forefront of this transformative field of drug discovery. We provide extensive expertise in the science of molecular glue and support the advancement of these compounds with our specialized research and development capabilities. I'm pleased to introduce our esteemed speakers of today's session, Dr. Helmut Buschmann and Dr. Anil Agarwal, who will share their in-depth knowledge and the latest insight into the development application of molecular glues. Thank you once again for joining us, and we're looking forward to an enlightening discussion. Helmut, stage is yours.
Thanks a lot, Dimitrios. Can you go for the first slide? Also from my side, a very warm welcome to the second part of the seminar series, Targeted Protein Degraders, with the today focus on molecular glues. If you can go to the next slide, I would give a short introduction and to wrap up what we learned during the first seminar. So the induced proximity targeted protein degradation is a groundbreaking strategy, as Dimitrios mentioned, in drug discovery that has emerged during the last 2 decades. Since 1999, the first publication was coming. So the TPDs, undruggable disease-causing proteins, are recruited for rapid destruction and elimination via the ubiquitin proteasome pathway, which is a major mechanism of protein degradation and homeostasis. So in the last 20 to 25 years, since the first small molecule was reported in the literature, the technology really moved from academia to industry, where several biotech and pharmaceutical companies have disclosed programs in preclinical and early clinical development. And several posters and presentations are shown in the MedChem congresses all over the world. Going to the next slide. We can still summarize that the TPDs are a major class that enable such proteins to be modulated through TPDs, are known as so-called proteolysis targeting chimera protein degraders, better known prodrugs. And what are the essentials of this type of molecules? These are 2 ligands joined by 1 linker. 1 ligand recruits and binds a protein of interest, the POI, whereas the other recruits a binding on the E3 ubiquitin ligase. And the simultaneous binding of the protein of interest and the ligase by the product induces then the degradation of the protein. And this degradation process is a catalytic process. So that means the product molecule as such is recovered and can enter in a new cycle again, which is a very efficient pathway. Just now, putting the learnings of next slide from the last seminar, Anil was trying to present you an overview about TPDs, the history, the starting points, the advantages of such new technologies, other modalities, milestones achieved in the last 20 to 25 years. However, also this leads to a quite competitive landscape, and he was able to provide us a deeper insight of this type of research. So the critical factors, challenges, and opportunities from the very early stages of the project on to bring it to the clinics, which is, of course, in many cases, a quite painful way. On the other side, also a lot of positive examples are now in the meantime reported in literature. So it was possible to provide very successful case studies in the development pipeline. And of course, talking about this type of molecules which have a certain molecular weight, 2 binding sites connected by a linker, of course, has limitations in physiochemical properties, stability, availability, and all these things. And today we would like to bring you to another area in this field, which is called the molecular glues. The molecular glues is, let me say, nothing different in principle, but it in many ways a further advanced strategy, which also interacts. The molecular glue interacts with two protein surfaces, whereas the E3 ligase as target protein and the protein of interest to induce enhance the affinity of these two proteins, just doing the same, but not with covalent or any other interaction, just by non-covalent interaction. And the molecular glues are monocovalent small molecules that chemically induce proximity of target of proteins with the E3 ubiquitin ligase to ubiquitate the degrade of specific proteins via the proteasome. So more or less exactly the same mechanism of action than the degraders and the product molecules we have learned so far. But of course, the interaction with a variety of target proteins at the same time may bring additional difficulties, because you need then affinity and selectivity to two different proteins within the same molecule or within two sides of the same molecule. But on the other side, you may have a lot of advantages with respect to molecular weight and so on. So it's not a surprise that molecular glues were really discovered by serendipity. It was not intended, and it was discovered using a molecule, thalidomide, which was called the monster of drug design, which was responsible in the late 50s and early 60s for severe birth diseases, but now again in pharmacies all over the world for severe cancer treatment. Bringing this to the next slide, this cartoon only should demonstrate that from the protein degrader concept, the products and the molecular glues are exactly doing the same. The only difference of a product is that you have two different sides of a molecule which are connected by a linker, and the molecular glue is only one molecule with different sides interacting with the same proteins at the same time, following the same catalytic cycle for protein degradation. So again, what we mentioned before, this type of advantage may lead to a better success rate in this field. However, it's still an early phase, and we will see what are the so-called advantages of the molecule. Just give you a short overview of what you will learn today in the next slide. That Anil, who will start soon with the presentation, will give us an overview of these proteins, and he will give us also a kind of introduction to the mechanism, to the milestone competitive landscape, to molecular glues, and of course also to analyze critical factors for rational design of molecular degraders. And of course, very important, how Taros could support the early starting point of such a project. So it's a fantastic approach to talk about the protein degrader. However, you need a lot of expertise to get the right starting points, because if you look to all the posters and publications, in most cases, these are describing unsuccessful results of such an approach. And the success rate really depends on the rational design at a very early stage for then a further optimization. So it will be now a pleasure to hand over to Anil, who will then present you more insights in the really fantastic world of molecular glues.
Thank you, Helmut, for that nice introduction to molecular glues. Really complex way of looking into drug development, but how nicely you have explained. So if I could just summarize what is molecular glues, which is obviously a new genesis after PROTAC. So when PROTACs came into picture, obviously there were lots of excitement, but with excitement, obviously there were lots of challenges, because PROTACs were obviously they were much bigger molecule. And it was difficult to kind of fine tune them to get into a smaller and a drug-like molecule. However, when molecular glues came into picture, that really came with lots of advantages, and we're gonna look into that today. So if I were to summarize, what are molecular glues? These molecular glues are, again, these are the targeted protein degraders, and they do this degradation by promoting or inducing protein-protein interactions. So basically, we'll look into the difference between PROTAC and molecular glues as well in a subsequent slide. But this molecular glue strategy has really opened up a new avenue in the area of drug development. And because there are many undruggable targets, and especially, for example, transcription factors or transition factors, which are very, very difficult to target or drug them because they don't have any binding site or they are really tough to target, we can still target them using molecular glues. And we'll see a few examples today how molecular glues are able to do those successfully. And if I were to name just few advantages of molecular glues, obviously these come with much smaller molecular weight. And with that, there comes lots of added physicochemical properties like higher cell permeability, improved PK/PD, and one of the greatest advantages of molecular glues is that it can degrade target proteins where it's really difficult to target with a small molecule because they don't have binding sites. For example, transcription factors such as G1/2S phase transition protein and similar other transcription factors we'll look into in more details in subsequent slides. Okay, on this slide, I thought to put together differences between molecular glues and PROTACs. I'm sure most of the audience are aware of it, but if I have to put for someone who's new here, we can see on the right-hand side cartoon, we can clearly see PROTACs are obviously much larger molecules because of the nature of it, because it needs to have a small molecule protein of interest part, you need to have the E3 ligase part and the linker. That makes a really bigger molecule. Whereas if you compare it with molecular glues, they are really like glue where it sticks the two proteins towards the two ends of the same molecule itself. So that's really the beauty of molecular glues. And if we look into the middle cartoon and the few differences, molecular glues, the difference between PROTACs and molecular glues are monovalent molecule, whereas PROTACs are bivalent. We don't need any linkers in molecular glues, whereas in PROTACs, we do need a linker to connect between the two protein of interest and E3 ligase ligand. Molecular weight mostly of the molecular glues, and we'll see lots of examples from the molecules which are in development stages. They are less than 500, whereas for PROTACs, it's more than 700 or 1,000 molecular weight. Most of the molecular glues, they fall under Lipinski's rule of 5, so they are much easier to develop as a drug molecule. Also, it works where you don't have even binding pocket, and we'll see those examples. So binding affinity wise also, even if you have very weak binder, molecular glues really works as magic. So now let's try to understand the mechanism of action. Although in general, all these degraders work the same way. Basically, they bring the protein of interest and the E3 ligase in close proximity so that they can be ubiquitinated and degraded. However, molecular glues work differently. So molecular glues doesn't have to bind to both the proteins at the same time. So in the cartoon on the top left, if you see, basically molecular glue binds between the protein of interest and E3 ubiquitin ligase. So by this, it allows ubiquitination of the target protein and degradation. But it does it by two different mechanisms. If you look in the bottom picture, you can see in cyan color effector protein or the E3 ligase, and in the purple color you can see target protein. One mechanism is it brings it induces the two proteins close to each other so that it induces protein-protein interactions and it stabilizes those interactions. By this, it brings the two proteins close to each other and those can be ubiquitinated and degraded. Other mechanism, which is one of the really important factor for molecular glues, is that they induce surface change or change in the surfaces of effector proteins or E3 ligases. And we'll see in more detail, this is the concept of understanding how we are able to do now a rational design for molecular glues, because we understand what are those changes on the surface of the effector proteins brought in by molecular glues and how it helps the targeted protein to bind to those effector proteins. We'll look into those details in the subsequent slides as well. So for now, in summary, we have two mechanisms. One is where the two proteins are brought close to each other and stabilize the protein-protein interaction. On subsequent one, molecular glues can bind to the E3 ligase partner, bring about changes in the surface so that it acts as a receptor and attract the target protein to bind to it, and then brings about the proximity and the ubiquitination followed by degradation. I hope that makes clear. Let me take you to the development history of molecular glues. I'm sure all the audience are aware of thalidomide, cerebron, and all all the news it made. So although thalidomide came to picture or in the news when the crystal structure of thalidomide with E3 cereblon was developed and produced in 2010 by Hiroshi Handa from Tokyo Institute in 2010. But thalidomide was in the news much before that as well. Actually, thalidomide was synthesized in 1954 by a German company called Grunenthal. And I'm sure people are aware thalidomide was given to pregnant women as a sedative because they were used to have morning sickness. But very soon people found that because of thalidomide, there were teratogenicity and kids were born with limbs defect. So in some countries, it was also restricted. However, this compound, this drug, was found to have lots of different other biological or pharmacological effects. In 1965, this was again so very good effect of this thalidomide was seen in case of leprosy patients. And then in the late 2000s, as you can see, this was approved for lots of multiple myeloma cases, which is kind of a blood cancer of bone marrows. So although it was approved, the mechanism was not clear how this binds. And this is when, in 2010, the crystal structure of thalidomide with cerebron was published. We knew it binds to the E3 ligand and then brings about the degradation of the important transcription factors of proteins which is involved in those kind of cancer tissues. After that, obviously, there was a huge number of development, and then as you can see, there are lots and lots of proteins were able to degrade, and most of these proteins are the difficult to target ones. So these mostly belong to the class of transcription factors or translation factors, which obviously are involved in lots of cell proliferation, survival, DNA repair, and all, and these are hard to target. So molecular glues not only is degrading just any targets, but these are degrading the targets which are difficult to drug or undruggable targets. And as you can see, from 2010 to 2020, many such molecular glues came into light, and we'll look into some examples in the next slide. In this slide here, you can see molecular glues in clinical development. Last week, we saw about a dozen compounds of PROTACs, which were in various clinical trials. And now here you can see maybe 24 compounds. They are into various clinical phases of development. And out of these, the first 3 drugs mentioned there in the list, thalidomide, lenalidomide, and pomalidomide, has already been approved by FDA for multiple myeloma. Again, this is kind of blood cancer caused mostly in the bone marrow area. So now let's look into a little bit detail on this table. If you notice, obviously there are more than 20 or 24 compounds in the clinical development. Out of these, 3 already approved drugs. And if you look into the second and the third column, you can see second column, it says all the ligases involved. Most of these are involved in the transcription and translation stage. If you look at the target proteins, again, these are again lots of different target proteins, from different RNA binding proteins to cyclin K. We can see now discovery. If you see strategy, most of the initial ones like thalidomide and all were discovered by chance, by serendipity. But over the period of time, as we understand the mechanism, there are lots of discoveries which is based on rational design, and we'll see what was the basis of that. Try to understand today. And on the property side of the table, if you look at molecular weight, SLOC-P, PPSA, and hydrogen bond donor acceptor, you can see most of the molecular glues fall under the Lipinski's rule of 5, and this is what makes these really exciting. Now, on the right-hand side of the picture, if you see, there are many big pharma companies which are already involved in molecular glues. Everybody knows about BMS and Celgene. Obviously, there are Novartis, Cipo Therapeutics, Monterosa, many more companies are coming in. And they're working not only in multiple myeloma, but with solid tumor and with lung cancer, which are one of the difficult ones to treat. Molecular glues are really helpful. Now, let's try to see a few examples of molecular glues, what they're doing and how they have been discovered. So the first 3 molecular glues, which were approved by FDA, if you look into these are obviously thalidomide, lenalidomide, and pomalidomide. Lenalidomide itself, in 2018 itself, because that's a published report, has made an annual sales of $9.2 billion US. That's a lot of money. just one drug, lenalidomide. So this thalidomide, as we just discussed, it was discovered as a sedative, but later on it was found that we can use it for multiple myeloma, and now we know it binds to cereblon. Similarly, the other two, lenalidomide and pomalidomide, these are also the most soluble versions or the derivatives of thalidomide. All these three drugs, they come under the category of, we call it immunomodulatory imide drugs. So these are used for multiple myeloma, as I said, and they all three recruit cereblon as a E3 ligase partner. Now, when we look at the crystal structure of these drugs along with cereblon, we understand that there is a nice pattern to which these follow. There's obviously a highly conserved tritryptophan cavity on cereblon, which obviously these make very nice interactions, and with that they're able to bring in the E3 ligase partner CUL4 and DDB1, which basically brings in the degradation of target proteins, which are the transcription factors as mentioned there, IKZF1, CK1 alpha, and SALT4. So this helps a lot, because when you understand how these molecules bind and what they're able to do, you can modify the structures, modify these molecular glues, and bring about the degradation of many more target proteins. Let's look at another example, which is there in literature, aryl sulfonamides. These aryl sulfonamides type molecular glues. So all of these compounds, as you can see on the left-hand side picture, they all have this aryl sulfonamide in it, obviously as the chemical name suggests. And these act as molecular glue by enhancing the protein-protein interaction between decaf15 and RBM39. Again, so RBM39 is the RNA binding protein, and decaf15 is again DDR and then CUL4 protein together. So they induce this degradation. And if you look at the right-hand side, see this decaf protein, which is shown in the blue and green color as the N lobe and the C lobe, and you can see RBM39 in between them. So what it does, these molecular glues, either Indisulam or E7820, if you see, they induce the protein-protein interaction between the decaf15, which is shown in the blue and green color, and the RBM39 in the pink color. So it brings in those two protein-protein interaction much closer to each other, and this is how then it allows for the ubiquitination of this RBM39 and then degradation. And if you see the hydrogen bonding diagram, you can see there are two very important interactions to be taken into account. One is obviously the hydrophobic interactions, as you can see, but the other very important is the water-mediated hydrogen bonding interactions, which is really crucial, which is shown in the crystal structure in the picture B above, and also in the cartoon diagram and on the top bottom right structure. So these interactions, when you look into the crystal structures, and then you can modify obviously this type of molecular glues, and you can bring the novelty into the structure. Now let's look into one of the very classical molecular glue degraders examples, which is there in literature about cyclin K degraders, CR8. Again, there's a nice story about the discovery of this molecular glue, which is obviously published in this paper, you can see, and the data from the paper. So basically in 2020, Benjamin Evert's group, they did this database mining. So basically they had 4,500 various clinical and preclinical anti-cancer drugs, and they used about 600 or so cancer cell lines. And what they did, they looked into the mRNA expression levels in about 400 of E3 ligases. And what they found is CR8, particularly the one of the enantiomer, it brings about the mRNA expression in this cyclin K, and then it was able to degrade it. And if we look into the mechanism, what we understand is a very unique way, and the medicinal chemistry, you love and enjoy it, how it binds. So basically, if you look into the bottom left structure, the B part, if you see, obviously this molecular glue CR8 binds to actually the ATP side of CDK12. When it binds to the ATP side of CDK12, and you can see the structure C, there is this phenylpyridine part, which is really important. And with the methylene bridge, it puts this phenylpyridine part into the pocket so that it brings the protein-protein interaction with the DDB1. Now, when this molecular glue brings in this protein-protein interaction between the CDK12 and DDB1, this now acts as a receptor and attracts cyclin K to bind on the opposite side. As you can see in the B structure, cyclin K in cyan color comes behind CDK12. So CDK12 and DDB1, they have a protein-protein interaction because of CR8, and this now allows cyclin K to bind at the proximity to CDK12, and this brings in so close to the DDB1, which is the E3 ligase, that the ubiquitination takes place, and the degradation of cyclin K. So basically, you have a molecular glue which starts binding to CDK12, but you don't degrade CDK12, you degrade cyclin K. This is the beauty of this molecular glue, and you can selectively degrade cyclin K via this molecular glues. Okay, so I mean there are many examples, and for the interest of time, we have restricted to few, and I can just show this slide here. There are many molecular glues in clinical trials, and I'm sure you're aware that in 2019, BMS spent $74 billion to acquire Celgene and its portfolio of molecular glue degraders. And you can see in this slide here, most of the compounds or molecular glues belong to Celgene, except for few, which are for C4 and Novartis and Eisai. Most of these compounds, which are either approved molecular glue degraders like lenalidomide and pomalidomide, and the others in the advanced stages of clinical trials. And maybe one day this can be a successful drug as well. Now, if you look at, so those were the clinical trials, and if you look at molecular glues in preclinical trials, that says that there's lots of excitement in this modality, and many big pharma companies and biotech companies are working on this area. And you can see on the left side, we have all these molecules which are into the preclinical highlights, and very soon they might be entering into clinic. Now, one important thing, if you look on the right-hand side of the slide, earlier we looked into, okay, degraders, maybe they mainly meant for cancer, oncology, and we saw in the last week that PROTACs not only does oncology treatment, but it's also applicable for HIVs. And now if you look at the disease indications, targeted indications, you can see, obviously cancer is one of the major part, but you can look into the immune disease, blood disease, infectious, neurodegenerative, and all the way inflammatory and metabolic diseases. So basically, all these companies are working on different therapeutic modalities by using molecular glues to their benefit. Now again, this slide I put here, taking from various companies' corporate websites. Molecular glues, the hidden treasure, as I had said to show you, there are lots of deals, mergers, and acquisitions. I'm not going to read all of it, you can read those, but if I have to read just in the last few months itself, if you see, the first one, C4 Therapeutics, it announced a strategic deal with Merck on the 4th of March only. If you look in Feb 2024, Novo Nordisk and Neoform, again, there's $1 billion deal. In Feb 2024, again, another deal with Vent AI and BMS. And the news goes on and on. And what it says that this big pharma company, if you see Merck, Novo Nordisk, BMS, Genentech, they are willing to spend money if the other biotech companies, for example, if you see C4 Therapeutics, Neomorph, or Proxygen, these. So what it says, what I'm trying to give the message here is, if you have a small biotech, if you have the passion and you have the ideas to design molecular glues and novel glues, I'm sure there are big pharmas who are ready to spend money and acquire those, and then so that you can do research for many more years. So now let's try to understand how do we do it. Again, in the interest of time, I won't be able to give you much detail, but there are critical factors which can help in the design of molecular glue degraders. Obviously, there are majorly two challenges. One is obviously, how do you select which E3 ligand to choose from out of 600 different E3 ligases? Obviously, most of the molecular glues have been working around cereblon, so that solves the problem, at least for now. Let's go ahead with the cereblon, and then it is somewhere I was reading the cereblon itself is able to target more than 2,500 of the human proteomes. So obviously, there's a lot of opportunities there. And obviously, once you have the E3 pool, and we know molecular glue, when it binds to E3, it causes the changes in surface that brings in the protein of interest to close to each other. So let's try to understand how that works. Okay. So what we understood so far, as molecular glues, they have the potential to degrade proteins that lack a small molecule binding site, which are normally undruggable, such as transcription factors with G2S phase transcription factors. We have seen example here. Now, molecular glues, we also know, they bind to E3 ligase, for example, cereblon, and then create a new surface that can recognize classes of proteins which with similar binding domain. And this binding motif on the E3 ligand or E3 ligase is referred to as G loop. Basically, these are 8 amino acid loop, starts with G as the must in the glycine, as you can see in other proteins in the middle picture. So there are 8 amino acid loop, which is called G loop. This loop is created when a molecular glue binds to the E3 ligase. Now, any proteins which are able to bind to this loop will be able to connect with the cereblon, and then you can able to degrade this protein. It doesn't end here. On the right hand side of the picture, you can see that obviously there is a thalidomide binding pocket, which refers to the thalidomide part which binds to the cereblon, which is shown in the left hand side, the third picture. However, there are again proximal and distal area. Basically, you can extend the thalidomide further to proximal and distal area, and that gives you extra efficient binding, selectivity, and affinity. So again, so there are lots of ways. You can see there's a reference quoted here. You can read for more details, but there are ways you can rationally design molecular glues now, and then you'll be able to develop a completely new molecular glue. So that brings me to almost towards the end of my presentation. A rise of molecular glues. You can see on the top A part, mostly it's shown as the PROTACs, where they don't obey rules of 5, Lipinski's rule of 5. They are bulkier molecules, whereas in the bottom part, blue color, you can see molecular glues. They're much smaller, much drug-like. And again, on the right side of the picture, you can see that is not just restricted, and the field, the area is just growing and growing and growing. And again, we are not restricting just to cereblon. Obviously, there are lots of E3 ligases, lots of interest in the VHL and other DTAP and other sorts of E3 ligands, which you can be targeted for different degraders. So in this area of interest, if anybody is looking to help, obviously we as Taros Discovery, Taros Chemicals will be able to help. Again, we have all sorts of services, but to name just the one which is relevant to today's topic, on the right hand side, you can see we have lots of experience in working with PROTACs and molecular glues. And one of our scientists is named as inventor in one of the clients that we were able to successfully give molecular glue. And again, in addition to discovery biology, we have all sorts of discovery, chemistry and discovery biology facilities from in vitro to in vivo, and we will be able to help if in any case anybody is looking to help. So thank you very much for joining these 2 seminars, and I hope you have learned something new, and it was really nice to have you here. Thank you so much for your time.